right, back to the salt mines then. All right, now a little bit more about eyes. This gets to the next exercise, which is 24. And so really I want to start on, in the older book, page 372. And in the newer one, it's actually about page 374. All right, now, first of all, on page 372 or 374, there's figure 24.1, which is the so-called blind spot test figure. Now, you notice that it has an X on one side and a dot on the other. Now, the idea is this. All right, if I'm using my right eye, I'm going to close the left, or else cover, so I'm just going to close it. And focus on the X as you move this in toward your face. And when I get to about here, the black spot has disappeared. Can you get that? Huh? All right. You have to get probably about 12 to 18 inches away from your eye, and the black spot should disappear. Now look at the X. Make sure you're looking at the X, and then as you move it in and out, and then as you move it in, probably 12 to 18 inches away, the black spot disappears. You no longer see it. Now, what's happening there is that, in essence, when you're getting the book at that distance away from your eye, you're getting the black spot right over the optic disc. And remember, the optic disc is the area where there are no photoreceptors, because that is where the optic nerve exits the eye. That's where the blood vessels enter and exit, okay? And there aren't any photoreceptors there. All right? Now, if you want to look at the left one, you really need to turn the thing upside down, and you do kind of the same deal. Look at the X, and when you get it in the right place, the black spot disappears. Okay? Oh, that's simple. All right. Now, some more terms to add to your growing lexicon. You know how I like to enhance your vocabulary. And you'll find these terms right there. All right, first one is emmetropia, or an emmetropic eye. All right? I think in the book it'll say emmetropic, but emmetropia is the condition, which is really good because it just simply means normal vision. Isn't that nice? Too simple just to say normal vision. So we have the term emmetropia. Now, we also have some other things that aren't going to give you nice, clear vision. And those are myopia, or myopic eye. Myopia is what's commonly called nearsighted, or nearsightedness. All right, now, what does it mean? Well, it means that close objects are clear, distant objects are not. Well, I have myopia. You're all blur now. I can't tell anything about you. And if I pick up the marker, I've got to get it right about there before it's really good and clear. But anyhow, supposedly that's the way it works. Now, why? Well, like they show in the diagram in the book, in myopia, the focal point is in front of the retina. All right? So in other words, the image, the sharp image is forming well in front of the retina, so therefore it's out of focus. All right? Now, how can you correct it? Well, one way to do it is use a concave lens like they show there in that figure 24.3. All right? Another way, of course, is with contact lenses. And remember now, contact lenses basically just reshape the cornea. Or if you want to go have that radial keratotomy stuff done, you know, where they have little thin slits in the cornea, you know, and that sort of thing, and that reshapes the cornea and that corrects your vision that way. Have at it. I don't think I care to do that. But anyway, there you go. All right, now, that is myopia. Now, we also have hyperopia. And that means farsighted, which basically means that distant objects are clear and near objects are out of focus. Now, in that case, as you note, the virtual focal point for the image is behind the retina. Well, you really can't get behind the retina, right? Because what's back there to stop light from going anywhere? The choroid, and not only that, the sclera will block it too. So, you know, yeah, the virtual focal point's behind the retina, but it really isn't going to focus back there. All right, and convex lenses take care of that. Now, there's one more, and it'll get mentioned in here eventually. I don't know why they didn't put it in here right now. Presbyopia. All right, presbyopia is a lack of accommodation. All right, now accommodation is the ability to focus on near and far objects. All right, ability to focus on near and far objects. So a lack of accommodation, well, the eye's gotten to the place it doesn't easily do that. All right, when you get to be about 40-ish, you typically develop presbyopia. And when you get to that point, you're either going to have to get reading glasses if you haven't needed them before, or you're going to get bifocals. All right? Because what's happening is the lens is becoming less elastic. It doesn't want to change shape to focus on near or far objects, and that's the whole idea there. It gets harder, it gets less elastic with age. That's normal. All right, now there's one more thing. I'll write it down here. I'll get this up. Astigmatism. All right. Now, in astigmatism, you don't have an even focus on the retina. And the reason for that is usually because the lens is misshapen. All right, it's not uniform in its contours. So, you may have a lens that, say, the bottom part of the image is sharply focused on the retina, which actually would be the top part, because remember it's upside down. But anyway, that part's sharply focused, but the other part is not. Or it might be the left half is sharply focused, the right half isn't, because the way the lens is not evenly contoured. And for that, you need to have a cylindrical lens to correct the vision. And it's like with my glasses. I don't know how well you can see that from here. But you notice with this right lens how much thicker it is on this edge than it is in the center toward the medial side. Well, that's because I also have astigmatism. So in other words, my lens is probably thinner on the lateral edge and thinner on the medial edge or something, and so, well, thinner on the lateral and thicker on the medial is what I should say. And so this is just the opposite to correct for that. And if you go to page 374 in the older book, or the newer one, you'll see it on page 376. There is the testing chart for astigmatism, and I'm sure you've all probably had this as part of your eye exam, where you have the spokes, like the spokes of a wheel, and you're asked, well, which lines are darker, So like with this one, it looks like 1 and 7 and 12 and 6, or maybe darker on that one, and this one's different ones, like 2 and 8 and so on. Okay, that's the test for astigmatism. Now, if they're all evenly dark, well, then you're going to have astigmatism, all right? But if you do, you'll probably see a difference in the shading on those lines. Okay.
Maybe.
And, huh? Okay, so we have all of that. And we're not going to use the ophthalmoscope or anything like that. We don't want to blind each other. And so we'll leave it at that, I think, for eyes. Let's see, I think I've got everything we wanted to mention. So let's go on now to some of the other special senses. And the next one, I think, is hearing. And that is exercise 25. And there we have the ear for hearing and equilibrium. And so here's a nice little model. All right, now if we start with the outer ear, on the outside you've got the auricle or pinna, as it can be called, and it's kind of a, sort of a, what would you say? Kind of a cone shape in a sense. I don't know. It filters, it concentrates. What is sound? What is sound? Huh? Yes, vibrations of the air, basically pressure waves. All right, high-low pressure. All right, and remember, like the original Alien movie, that series, what the subtitle was: "In space, no one can hear you scream." Well, now why not? Of course, you couldn't breathe there either. But anyway, well, there's nothing to, take care of the sound. Sound does travel underwater, too. In fact, actually better than it does in air. But in any event, that catches the waves, the pressure waves, and directs them through this opening in the temporal bone. What was that opening called? That'd make a lovely question again. We had it once. External acoustic meatus, absolutely. And it comes right back to this little number called the tympanic membrane. And on the external surface, that's covered with skin, and on the internal surface, it's a mucous membrane. Now, the tympanic membrane, just like it shows you there in the book on figure 25.1, is kind of cone-shaped with the apex pointing in, all right? And when a pressure wave of sound hits that thing, it causes it to vibrate. So the high-low pressure waves cause that to move in and out to actually vibrate. Now, as that vibrates, then we're into the middle ear area. And in the middle ear, we find the three what are called auditory ossicles. And the auditory ossicles are three little bones, all right? And they are the malleus, the incus, and the stapes. And I will pass this giant economy size model around here a bit. So here is the tympanic membrane, and attached to it is the malleus. As the tympanic membrane moves, as it vibrates, it sets up movement in the malleus, which in turn causes the incus to move, which in turn causes movement of the stapes. Now, the stapes means stirrup. It's a stirrup-shaped bone, and you can notice that on here. And it acts a lot like a piston. So as the tympanic membrane is vibrating, it's causing that stapes to move in and out on an area of the inner ear called the oval window. And more on that in a bit. All right, so here we go. And if this one thing, now it's going to fall out, I'll just take it out. There's no sense of it fall out. For some reason, it's scripted and doesn't want to stay in there anyway. All right. And on this model, we have the same thing, it's just smaller. So they have the malleus and the ancus attached, and then the stapes part of it is here over the oval window. All right. So there we are. Now, one other little thing that we have, and it's this structure right here, which is a tube-like structure that goes from the middle ear down to the pharynx, down to the throat. And that can be called a number of things. The old-fashioned name is eustachian tube. It can also be called the auditory tube. And your book calls it the pharyngotympanic tube. Any of the three will do. All right. Now, what's it there for? Well, it's there to equalize pressure on either side of the tympanic membrane. Because if you didn't have that feature, what would happen? All right, high pressure wave comes in here, presses in on the tympanic membrane, builds up pressure inside here, you've got a nasty earache. All right? And of course, as you well know, and some of you may have experienced this, I don't know, I didn't, thankfully. If this thing gets blocked or occluded, and it happens a lot in growing children, they have earaches. And how do you remedy them? The tubes going through the tympanic membrane, so that helps to equalize the pressure on either side. Okay? You know, and this is also the bit about, you know, when you're in the plane and it's taking off and they haven't quite got the pressure regulated inside your ears. Pop, you know, when you swallow and all that kind of stuff. Or driving over the mountains and that kind of thing. Okay? All right, so we have that. Now, the inner ear. Well, the inner ear is really for two things. It's for both hearing and it's for equilibrium. Now, where we find the oval window, where that stapes moves in and out, that is an area of the inner ear called the vestibule. The vestibule. All right? And when it comes to the vestibule, there are two real functions for it. And if you, let's see, I think I've got a chart here that tells us this stuff. They don't. Okay, now here's where the new book is better than the old one. Figure 25, table rather, 25.1, talks about the different areas there of the outer ear and the middle ear and inner ear and that sort of thing. And I don't see that table in this older version of the book. So if you don't have it, we can make you a copy of it. Just let me know. All right, but here's the deal. Vestibule. It's for both hearing and it's for what is called static equilibrium. Static equilibrium. Now, what does that mean? Well, static equilibrium is the kind of equilibrium that is when you're sitting still so you know the position of your head in space. All right? So in other words, I can close my eyes and I know I'm looking straight ahead even though I'm not looking at anything. Or I know I'm looking down, or I know what I'm looking up, or something like that. But I'm not moving. All right? That's static equilibrium. And the organs in that vestibule area are responsible for that. Okay? Now, we also have this snail shell looking structure. That is called the cochlea. All right? And it actually comes apart on that model so you can see inside. It does on this one too. The cochlea is for hearing. And that is where the organs of hearing are going to be located. A little more on them in a bit. All right? So that snail shell looking thing, that's the cochlea. That is for hearing. Now, the other things, the three other semicircular canals, as they're called. All right, and you notice that they're all at right angles to each other. Those are for dynamic equilibrium. Dynamic equilibrium. So as you're moving, the brain can figure out where your head is in space compared to the rest of your body. And of course, one problem that you get into is if you go into a quote-unquote weightless environment, that's one reason why people can sometimes experience vertigo, which is a hallucination of movement, in that kind of a situation, because there's no gravity acting on that stuff. And what's in there? Well, we'll get to that here very shortly. All right, so the semicircular canals are for dynamic equilibrium. The vestibule contains the organs that are for static equilibrium. Okay? Now, let's say a little bit about the cochlea. And if you will flip to either figure 25.3, and it's either on page 386 at the top or 386 at the bottom. They finally got the pages caught back up again. All right, page 386, either at the top of the page or the bottom of the page. There you will find, in essence, the areas within that cochlea. And they are filled with fluid. And it's either going to be perilymph or endolymph. Now, as that stapes moves in and out like a piston on that oval window in the vestibule, it's going to cause those fluids in the cochlea to start sloshing about, if you will. It puts them in motion. And that motion of those fluids is what will be detected by what are called hair cells. The hair cells are the cells that will actually generate the nerve impulses that go off to the brain for interpretation as sound. All right? So, the movement of the stapes sets the fluids in motion. Those vibrations of the fluid picked up by the hair cells that will, in essence, generate the impulses that go off to the brain for interpretation of sound. Now, where is sound really detected? Well, the spiral organ. You see that labeled there on the diagram? Spiral organ. All right, those hair cells are in there. Another name for the spiral organ is the organ of Corti. It's C-O-R-T-I. Probably wouldn't hurt if you remembered both of those, because since your instructor is a geriatric individual, that's the one he likes to use because he's old-fashioned, you know. All right, so there we go. The organ of Corti or the spiral organ, either one. That's where the hair cells are located. That's where the nerve impulses are generated then. That, in essence, will be interpreted as sound. All right? So that is in the cochlea. All right? And the semicircular canals are there for detecting movement. So we know where our head is in space when we're moving. That's, of course, called dynamic equilibrium. And so we may have a little more to say about that in lecture after Thanksgiving. All right? Now, another set of special senses: odor, olfaction, and taste. And those are in exercise 26, which begins on either page 398 or 400 are the pages that I want to look at in the book, depending on which version you've got. All right. 398 and the new, and no, the older version 398 is 400 in the new one. Okay. find it? All right, let's deal with olfaction first, because that one's one that's easy to deal with, because we've already talked about a bunch of that stuff before. Now, where is the olfactory mucosa? Well, it's in the nose, in the inner nose, and it is along the superior border of that superior meatus, okay, within the nose. And remember now, the cribriform plate of the ethmoid bone had all those little foramina in it that the nerves, the olfactory nerves, pass up through to form the olfactory bulb. Remember that? From the brain when we talked about that. All right. Now, the detection of odors. All right. If something is going to be detected odor-wise, it's got to be a gas, so you can take it in when you inhale. It also has to be water-soluble, because that mucous membrane has a thin sheet of mucus over it, and mucus is mostly water. So it's going to have to go in solution in that. And then, at the ends of those olfactory neurons, which are bipolar neurons, remember, there are what are called olfactory cilia. Now, even though they're called cilia, they don't move. These are more like, if you will, more like microvilli, actually. And embedded in the membrane of those olfactory cilia are receptors, proteins that act as odorant receptors. Now, there are approximately 500 different unique odors that you can detect. And so when you're smelling something, it's probably not just one of those. It's probably a combination. Okay? So the odorant molecule is up to 500 different things that you can detect odor-wise. All right? They come in, they combine with the receptor. That combining with the receptor is going to generate an action potential in those bipolar neurons. Remember what that means. And off goes the impulse up to the olfactory bulb, and then to the olfactory nerves, back to the olfactory cortex of the brain. All right, where you can interpret it. And, you know, like with other sensations, you can actually remember odors. You know, you can have the sense of them even when you're not smelling them right now. Another interesting tidbit, I think, about odors is the fact that perfumes and colognes are definitely complex mixtures. You don't want something that is all just like rose oil, you know, some really pleasant smelling thing, because it would be kind of flat. And so perfume makers put in some kind of caprylic ingredient. Caprylic comes from the Greek for goat, so you can imagine what that means. And it's just a teensy little bit, I mean, not enough that you can smell it, of course, but without it, it would have a flat, you know, your perfume or cologne would just have a flat sort of smell to it. You don't want that. Do you? I mean, that's why you spend all the big bucks for that stuff. Right? Okay. I realize a little dabble-do now. All right, now, there it is. There's that superior nasal meatus. Now, what happens if you've got a cold? What happens? What about your sense of smell when you have a cold? It's dulled, isn't it? Now, why, I wonder? Well, what happens to that nasal mucous membrane when you've got a cold? It becomes inflamed. It becomes swollen. There's also a hypersecretion of mucus. What do you call that stuff that comes out when you sneeze and you got a cold? Snot. Exactly. That's what it is. Excess mucus. Well, remember now, odorant molecules are going to have to diffuse through all those extra layers of stuff if they want to get to the olfactory cilia and be detected. So, obviously, when you go bulking up the membranes, you go adding a thicker than usual mucus sheet, well, it's going to be harder for odorant molecules to diffuse through to those olfactory cilia. All right, there you go. Now, what about taste? Well, the gustatory sensation, taste. Well, the tongue is key to it because on the tongue, what have we got? We've got those papillae. And the papillae are those little elevated areas on the tongue. And on the side of those are taste buds. Now, not all the taste buds are actually on the tongue. That's where the bulk of them are. There are a few back on the pharynx. Not many, however. All right, now what is a taste bud? Well, if you look at the diagram, figure 26.2. It's on either page 399 or 401. All right, you see the papilla. You see where the taste buds are there on the side. And then if you look at the diagram for the taste bud itself, you'll notice that there is a taste pore, a little opening, a little hole, that allows material to enter and exit. So anything you're going to taste is going to first have to be dissolved in saliva. So it's going to have to be water soluble, right? Because saliva is mostly water. All right, so that material then diffuses in through the taste pore into the taste bud. And there in that are taste hairs that are, and they're calling them gustatory hairs in the book, but taste hair is another name for them. And those are an extension of what are called taste cells. And the taste cells are actually a modified epithelial cell. And again, there are receptors embedded in the membrane of those taste hairs that detect the basic tastes. Now, there may have been 500 basic odorants, but there are really only about 4 or 5 and maybe 6 or 7 basic tastes. The jury's still out on this. We've known forever there are these 4. Sweet, salt, sour, and bitter. Now, most folks will agree that a 5th one is umami. All right, so there are at least those 5 that I think everybody can agree on now. All right, another one that some people suggest is probably a real taste is metallic. Okay? But at least there are those 5. Sweet, salt, sour, bitter, umami. Now, what on earth is umami? You're all probably saying, what the devil's that? Well, meaty, or it's Japanese is the word. It's meaty or, yeah, my mind just went blank. Savory. Okay. Meaty or savory. The purest umami flavor is that of monosodium glutamate. MSG, the food additive. You know, I think it's accent. That's largely what it is MSG. Okay? You know, it's kind of, it heightens flavors, you know, but it's thought to be a basic taste. Well, tastes, again, are mixtures of these basic tastes. You know, and the example I like to use is that my one grandmother was ape about that candy corn. She always had that stuff at Halloween, Thanksgiving time. I never could stand it because to me it was just sickening sweet and tasteless. I mean, oh, it had taste, sweet, yeah, that's it. Well, you know, if you're baking cakes, cookies, pies, whatever, you always put a pinch of salt and things, right? Because that tends to help, you know, heighten the flavor. You know, you don't want things that are just all sweet. And while, you know, salt's nice, you don't want just salt, all right? You know, you do want some other things in there for the most part. So, most pleasurable flavors are mixtures of things. Bitter is actually thought to be, you know, anything that's purely bitter, it makes you shudder. Probably because most poisons, like a lot of alkaloid poisons, are bitter. And that was probably, you know, a sort of an evolutionary sort of thing, you know, that helped us distinguish things that we probably shouldn't eat, you know, that sort of thing. But a little bit can help enhance the taste. I mean, you know, like the bitters that you put in certain cocktails and that sort of thing. You don't want a lot. You want just a little bit. You know, there you go. All right, so there we are. And that, in essence, is really about all there is to taste. So, if we have then the flavors linking up with their receptors in the taste hairs, or the gustatory hairs of those gustatory cells, that will cause those taste cells to generate an impulse that goes off to the nerve and off to the cranial nerve. It carries it off really to the brain for interpretation of flavor. And actually, as we're going to see when we talk about cranial nerves, there are two different nerves. There are ones for the anterior 2/3 of the tongue and another one for the posterior 2/3 of the tongue. And once upon a time, people talked about taste zones. That's pretty much passé. It does appear that all these basic tastes are pretty much scattered throughout the taste buds. At one time, it was thought, the sweet was on the tip of the tongue, and then bitter was in the back of the tongue on the posterior side. But that's gone by the boards, I guess we could say. All right, so we may have a bit more, and we probably will, to say about that in lecture, but not a lot more. So there you are with a little intro to that. And I'm not done. Now remember, we have the diagrams. We haven't gone over those yet. No, I didn't give them to you just as coloring book exercises. There's the eye, right, and we have the spinal cord, and we have two views of the brain, right? Got them? Did you forget yours? All right. Well, let's take a look at these. All right, now, let's start with the eye. I like that one first. What is number 1? Huh? Cornea, right. Now, number 2. All right, number 2 could be two things. It could be the space, or it could be the opening, all right, within the iris. So what would you call it? If you're considering it the space is filled with a watery fluid, it's the anterior segment. And if you're considering then the opening through the iris, it's called the pupil. All right. Number 3 is definitely anterior segment, is really what that's referring to. Now, number 4, any bids on that one? Ciliary muscles. Okay? Ciliary muscles for 4. All right, number 5. All right, that would be the innermost lining or tunic, if you will, in the eye. What would that be? Retina, correct. Number 5 is retina. Number 6, choroid, yes. Number 7, sclera. All right, now number 8, that little indentated area, fovea centralis, okay? And remember, what are the photoreceptors that you find present in very high concentrations there? Cones, yes. And 9 is referring to the blood vessels. So what have we got? Central artery and vein, right? Okay. Number 10. That's the optic nerve, that's right. Okay? Because the blood vessels, remember, are in the center of the optic nerve. Okay, then the nerve fibers are all around it. All right? So number 10 is actually optic nerve. All right, now number 11. Generic. It's an external muscle, so what would we call it? Extrinsic eye muscle, yes. Which one? Who knows? We don't know what axis we're looking at here. All right? So it's an extrinsic eye muscle. Number 12. Yes, ciliary zonules, correct. Number 13. Iris. And number 14, that's the lens, yes. Now, simple is it not? Yes. All right. Go ahead, it's on this side. All right, spinal cord, I like the spinal cord. Next. And these are lettered instead of numbered. All right, A, what's that hole in the middle called? Still the cerebrospinal fluid in life. What's it called? Central canal, right? Yes. A is the central canal. Now, B. Now, which side of the cord are we on? Anterior or posterior with B? Posterior. And that's a white matter column, so it's a posterior column. All right? Posterior column. All right? C is a gray matter horn. Which one would that be? No, it's not C. Toward the back, so it's posterior. Posterior gray horn. All right. What kind of information passes through that? Sensory, yes. D is lateral horn. Now, we haven't talked about that yet. That's going to figure in when we talk about the autonomic nervous system. We'll chat about that in lecture after the break. Okay? All right, that's a lateral gray horn, and it's actually part of the autonomic nervous system. All right, now, E. Ventral root, yes. And what kind of information passes through that? Motor, yes. All right, F. Now, the two roots have come together before you get to F, so what would F be? That's the spinal nerve. That would be the spinal nerve where it emerges from the vertebral foramen. Okay? G, that enlarged area there, what would that be? Dorsal root ganglion, correct. And what is located in the dorsal root ganglion? The cell body of which kind of neurons? Sensory or motor? Sensory neurons. And what kind of neurons are they? Unipolar, bipolar, or multipolar? Unipolar neurons. Very good. All right. Now, H. Now here, remember, if we get to H and I, the nerve has emerged from the vertebral foramen, and it's now split into two rami. So what is H? Well, I think it'd be the dorsal ramus, don't you? Where's it going? To the back. Muscles and skin of the back. What kind of fibers are in it? Sensory or motor? Bless you. Both. Because remember, spinal nerves are mixed nerves. They have both sensory and motor fibers in them. All right, what's I? Ventral ramus. Yes. Where's that one destined for? Well, it's going to go to the muscles and skin on the lateral as well as anterior side of the body wall, right? All right. J refers to the covering. What's that outermost covering called? Now, what's the outermost covering on the brain called? Huh? Dura mater. Well, guess what it's called on the spinal cord? Dura mater. Same thing. Because remember, those same coverings that you find on the brain come right down through the vertebral canal and cover the spinal cord as well. All right, K. What's the middle layer called? Or meninx, if you will. Arachnoid, yes. And then L is the Pia mater, right? The delicate mother, the one that's most intimate, the one right on top. All right, now M, what's that gray matter area called? Anterior horn, right? Okay. O, that area of gray matter that connects the right and left sides is called the central comma shear, right? Remember that? Oh yes, we talked about that. And N, forgot N, what would that be? Huh? Well, you could just say that it's a dorsal or posterior nerve, and it's or sensory nerve, either one, something like that would work. Okay, there we go. Very good. Now, the brain. Well, let's start with the inferior aspect. That's number one. All right. What is number one on the diagram? They saw that one on your sheet brain very prominently. Olfactory bulb, yes. Number two. What's it? Optic nerve, is it not? Yes. Now, number three. If you miss this one, you ought to get 20 lashes at least. Optic chiasma, absolutely, excellent. Number four. Now this is posterior to the optic chiasma. We just talked about that today too. Called the optic, starts with a T, tract, optic tract, right? So you have the optic nerves, they come together with the optic chiasma. Some of the fibers cross, some don't. So when you go posterior to the optic chiasma, they're called optic tracts. All right? Number five, what lobe of the brain is that referring to? Temporal lobe. All right, now number six, that enlargement there on the, back here, let's pick this one up. Right there, what's that called? That enlarged area. Starts with a P. Pons, yes. All right. Number seven, what's this? Medulla oblongata, right? Okay. Number eight, cerebellum. Now, let me just taunt you a little more. What are the gyrations on the surface, the gray matter of the cerebellum called? Stack of leaves. Folia, yes. I thought I heard somebody say it. All right, number nine. Well, it would be an olfactory nerve or olfactory tract. Either one would work for that. Okay? All right.
What was number 8?
Cerebellum or folia if you're thinking about the folds, all right, in the gray matter. All right, now let's go to the last one. That is the sagittal section of brain. All right, number 10. What lobe is that pointing to? No, not occipital. Parietal, right? 11 is occipital, okay? See? All right, because see, like here is that central sulcus. So there would be the, well, I don't want to give that away because we have that later too. All right, so 10 is parietal, 11 is occipital. Number 12. Now, what do you think that is? Now, remember on the sheep brains, we had to bend the cerebellum down out of the way to get to it. No. I mean, it's in there. It's in that same general area. But this would be more like the corpora, quadrigemina. Remember those? Yes. All right, number 13. Now, what are the tracts in there called? The white fiber tracts. Arbor vitae, yes. All right, and of course, they're found in what part of the brain? You already had it. Cerebellum. All right, now number 14. Well, we've already mentioned this one before, too. Medulla oblongata, right. Number 17 we've had before. What was that called? Pons, yes. Number 15. Oh, shame on me. I forgot 15 and 16. Now, what's 15? Now, that's a gland. We saw it on the sheep brains where we peeled off all the meninges. Pituitary gland, yes. What is the little stalk that it hangs from? What was that called? We found that, actually, the remnant of it on the sheep brains. Starts with an I. Infundibulum, right. Now, number 16 is something we really haven't talked about yet. We'll chat a little more about this one in class after Thanksgiving. Hypothalamus. All right, number 16 would be referring to the area called the hypothalamus, which is below the thalamus. All right? And it's going to play a key role in a lot of different things. And it also plays a key role in regulating that pituitary gland, too. All right, so 16 would be the hypothalamus. What's 18? Well, the 18 would be white fiber tracts that connect the two hemispheres of the brain. Corpus callosum, right? And number 19, well now, what lobe of the brain is that? Frontal lobe, right. And what bone covers the frontal lobe? Frontal bone, yeah, that's how they get the name, of course. All right, now there you go. Well, I think you suffered enough. But now you did enjoy the eyes, I know. All right. Well, I thought that was fun, didn't you?